Thank you.
Let me turn now to Dr. Adam Zlotnick, who's the Distinguished Professor of Molecular and Cellular Biochemistry at Indiana University, where he's done a lot of work on the core protein of hepatitis B virus, and will be giving us an overview of capsid assembly modulators. Adam?
Thank you very much. I hope you can hear me.
Yes, I can. We can. Thank you.
And let's make sure I have a pointer. Right. Thanks very much for giving me the opportunity to talk about capsid assembly modulators. Here are my disclosures, and the take-home is that I am an enthusiastic and emphatic advocate for attacking the core protein towards therapeutics and an eventual HBV cure. The 1-minute version of this whole talk is that there are 4 mechanisms of action for CAMs, capsid assembly modulators. 3 of them are based on the fact that the HBV capsid protein, HBC, has structural activities. 1, 2, 3. 1, CAMs are assembly agonists, leading to empty and defective capsids. 2, CAMs bind capsids and can dysregulate disassembly. 3, CAM-induced misassembly can lead to innate immune stimulation and apoptotic destruction of infected cells. HBC also has non-structural activities, and CAMs appear to be able to modulate these as well, making a 4th mechanism of action. The star of our show is the capsid protein, HBC. It's a small dimeric protein with an assembly domain and a basic C terminus. Purified or in cells, it can spontaneously assemble into capsids, predominantly a T=4 120 dimer capsid. And notably, the vast majority of these capsids in cells and in infections are empty. And so when a CAM induces assembly of an empty particle, it's not creating something new, it's following a well-established biological pathway. Jae-Ming went over the viral life cycle. I just want to point out that CAMs and HBC have roles in just about every step of that life cycle. CAMs in particular have roles in disassembly, assembly, and in the nucleus where we have cccDNA. How do CAMs work, particularly as assembly agonists and capsid disruptors? Well, the capsid is built of these dimers, and you can see 2 dimers here highlighted and here blown up. In the light blue dimer, we have a groove right through over here, and the dark blue dimer fits directly into that groove. This red density over here is actually a CAM molecule. It sits in a pocket in the groove, increases the amount of buried nonpolar surface area, and thus increases the association energy. And it can also modulate the protein-protein angles affecting the assembly products. All known CAMs fit quite nicely into this pocket. Here you see in the apostructure, the groove and the CAM binding pocket. And here you see a dibenzothiazepine, a HAP, a sulfamoyl benzamide, and so on. Alternative sites have been proposed for CAMs, and you saw one alternative site identified by at the dimer interface, identified by Zhou Wang, working with Jae-Ming Hu, and also independently by Anya Bachman and Michael Nassel. So what do I mean by inducing assembly? This is purified protein over here, and we can induce assembly simply by adjusting solution conditions. We're looking at light scattering. Big things scatter a lot of light, small things just very little. After we induce assembly, we get an increase in light scattering. The more CAM we have, the more light scattering we get, and the faster it appears. Quantifying these same reactions using the same color scheme, by size exclusion chromatography, we see that the more CAM we have, the less dimer we have, and the more capsid and non-capsid polymer. CAMs have been subdivided into CAM-Es that lead to empty capsids and CAM-As that lead to aberrant assembly. In a normal assembly reaction, you come up with a large variety of, actually a large collection of very regular particles, and that regularity is emphasized in these class averages. In the presence of a CAM-E, we should expect more of the same. However, and this is a classic CAM-E, sulfonyl benzamide. However, in class averages, we can see that the vast majority of these particles are actually defective. They are no longer circular. This is nothing nearly as florid as what a CAM-A will do. A CAM-A, like a HAP, favors hexagonal sheets and can lead to huge, monstrous things. Dibenzothiazepines have been identified as CAM-Es and CAM-As, depending on the author, really. But one can see that excess DBT1 leads to tubes and conical particles. So this classification has to be qualified. Cryo-EM shows that CAM-Es lead to defects not easily seen by negative stain, and CAM-A defects are not always apparent when the CAM-A is added at a low dose. So this first mechanism of action, CAM-induced assembly, these are the first generation CAMs. Some of them date back into the late 90s. The two best of these have been shown to have wonderful therapeutic activity, but not be curative. Such molecules are easily discovered and modified, and they can yield normal and aberrant morphology, but the difference between those two morphologies is a little vague. Second mechanism of CAM activity really was developed in this paper by Jan Martin Burke and has since been advanced quite nicely. What was shown was that there's a real difference between the behavior when CAMs are added in the first, immediately after assembly versus after a few days when the infection has had time to establish itself. Under both regimens, the two CAMs investigated were effective at suppressing production of new virus, but the effect of the regimens on production of other biomarkers, notably viral RNA, the E antigen, and the S antigen, were quite different. In an established infection, highlighted in this upper box, neither CAM really had any discernible effect on production of the biomarker. However, when CAM treatment started at about the same time as infection, both CAMs were shown to suppress biomarker formation. Apparently, in mechanism of action 2, CAMs prevent new infection. This effect has to be started at the time of infection, and it apparently works by the ability of the CAMs to melt the infecting virus. And that's really does seem to be the case. Here we're using DBT, one of my favorite molecules, to disrupt capsids. When DBT is added to capsids at low concentration, there's really no effect. But above a certain threshold, capsid disappears and subunits and dimers become the dominant species. I should have noted this is a size exclusion chromatograph. And some work from Assembly Biosciences with a highly active CAM following mechanism of action 2. This is actually work by Narudin Anshwanawala. The molecule was tested for its ability to disrupt a capsid and expose its DNA to nuclease digestion. At low concentration, not much happens, but above a threshold, CAMs seem to break up the capsid, leading to destruction of viral DNA. The first-gen CAMs had mechanism of action too, but it wasn't particularly active with the EC50 in the micromolar range. Current generation of CAMs now have 20 nanomolar and lower EC50s for this mechanism of action. Mechanism of action 2, CAM-induced disassembly, is related to assembly. It probably works by stabilizing bad geometry, leading to a rupture of the capsid. And second generation CAMs emphasize both of these mechanisms, disruption and assembly. A third mechanism of action is that CAMs apparently are able to lead to an apoptotic event. Does A stand for apoptosis? This is work that was originally demonstrated by the Oligos group and collaborators. And what they observed is that prolonged treatment with a CAM-A had remarkable effects. The CAM-A led to accumulation of nuclear puncta. And in this mouse model of HBV, a prolonged treatment, well, initial treatment led to a decrease in HBV DNA production. And after a lag phase, there was a loss of S antigen and E antigen. And that loss of these biomarkers was concurrent with the appearance of an ALT flare that and correlated with a evidence of apoptosis through a tunnel assay, tunnel staining, and proliferation of uninfected cells. There's a relatively small literature on these apoptotic effects. And in summary, we see that the apoptotic effect is related to aberrant assembly localized to nuclear puncta. Not all CAM-As are created equal, and some seem to be more effective in driving apoptosis than others. The antiviral effect, or the apoptotic effect, requires relatively high levels of HBC, and one can see that without that, one won't have much of an aggregate. And I would anticipate that in integrated genomes where there is no HBC formed, this effect is not going to be very obvious, if at all. The fourth mechanism of activity is the effect of CAMs on cccDNA expression. While it's easy to pick out a starting point for the observation of mechanism, for the first observations of the first three mechanisms, as soon as CAMs started appearing, there seemed to be some evidence for these other activities. Now, in earlier, I showed you that CAMs act by binding at a dimer-dimer interface. For CAMs to have this alternative activity, one would expect a CAM to bind to an individual dimer. And recent work from my lab in collaboration with Brian Bothner's shows that when CAMs are added to free dimer, not capsid, it led to an increase in hydrogen deuterium exchange across the molecule. That is, that the hydrogen bonds that are holding alpha helices together are being made or broken at a faster rate in the presence of CAMs than in their absence. Conversely, if you add CAMs to capsids, it slows down the rate of HD exchange. A bit over 10 years ago, Massimo Livrero and collaborators showed that HAP molecules, a CAM-A, were able to affect HBV epigenetics, both in a transiently transfected cell line or in a cell line that is stably transfected and able to accumulate cccDNA, samples were chipped down and tested for epigenetic markers. Notably, acetylated histone H3 decreased and the amount of bound HBC decreased. My lab, in collaboration with Door Pharma, has identified a subset of molecules that are able to suppress virus production and also S production. And this particular molecule was presented at the HBV meeting in 2021 at DPO4 167. Evidence of CAM effects on cccDNA are present in the literature, sometimes in unexpected places. The goal of this very recent paper by Taverniti et al. Was to examine CAM-A-dependent core aggregation and apoptosis. They used a CAM-A as a control molecule. Interesting, as expected, both the CAM-A and the CAM-A were very good at suppressing viral DNA production, but unexpectedly, both the CAM-A and the CAM-E effectively suppressed S production. For the CAM-A, this is correlated with apoptosis. For the CAM-E, which had no effect on viability, the mechanism is in question. I would suspect it may be a transcription-dependent effect. So mechanism 4, CAMs can act on isolated dimers. CAMs can modulate expression of HBS. CAMs can affect epigenetic markers. The details of this mechanism from CAM to CAM are not known, and any optimization of this mechanism is still at a very primitive level. So in summary, I'm sorry, I'm going a little over time. In summary, there are 4 mechanisms of action: assembly, disassembly, induced apoptosis, and action on cccDNA. CAMs have been optimized for assembly and have completed phase 2 trials. They've been shown to be therapeutic, but not curative. CAMs have been optimized for disassembly, and these are now in clinical trials. In the future, I look forward to studies of apoptotic induction and action on cccDNA. And thank you very much for your time.